In February 2016, the Human Fertilisation and Embryology Authority, the HFEA, approved a request by the Francis Crick Institute to use CRISPR-Cas9 gene editing on human embryos. And although the embryos were not allowed to develop beyond 7 days, this was the first time a regulatory authority had granted such research anywhere in the world. In December 2016, the HFEA made another controversial decision by allowing clinics to seek licenses to perform mitochondrial replacement therapy, or MRT, to cure mitochondrial diseases. As the embryos that receive MRT will be allowed to develop without being terminated, and although the nuclear genome is not affected by MRT, MRT is often viewed as a gateway to gene editing in humans, as new genetic information is supplied to the host. Based on your opinion of genetic modification in humans, MRT acting as a gateway to gene therapy can be viewed as both a good or bad thing. An argument that stands both for and against gene therapy in humans is that germline editing is permanent and is passed on to subsequent generations. Therefore, if a human that goes on to reproduce has their genome modified, so will all future generations that come from them. The three main techniques used in germline editing of mammals are zinc finger nucleases, TALENs, and CRISPR. As you are already aware of the biological processes each one uses, let's talk about the advantages and disadvantages. CRISPR is currently at the forefront of gene editing due to its potential, but also as it is relatively the simplest technique and most efficient, as the scientist does not have to create a recombinant protein specific to the genome, like with zinc fingers and talons, but rather just a guide RNA sequence. However, a disadvantage is that as the guide RNA sequence is much more simple, this leaves scope for more off-target effects, but we will talk more about those later. A question raised is how best to get the CRISPR complex into the cell or embryo. There are 4 main methods to do this: plasmid transfection, direct delivery, transfection of mRNA encoding Cas9, and viral vector transduction, which you will have covered in lectures. Zinc finger nucleases, or ZFNs, were the first of the 3 methods to be created and have a fairly high level of site specificity, as the recognition protein that is used to guide the complex can contain many peptides, which recognize the DNA bases in triplets. Also, unlike CRISPR, these work as a dimer, which increases site specificity even further. As TALENs use single nucleotide recognition rather than triplet recognition, they are easier to design than ZFNs and still maintain a high level of specificity, reducing off-target effects. However, TALENs are still not as simple as CRISPR to design. Furthermore, TALENs are able to cut DNA at more sites than ZFNs, as the triplet binding of ZFNs limits their use. Electroporation is usually used to introduce them to the cell. All of these techniques are still used today, but in terms of a creation timeline, ZFNs came first, then TALENs, followed by CRISPR. Now to move on to some more of the ethical arguments surrounding gene editing in humans, and to try and help, I will be using green for arguments in favour of editing, red for arguments against, and black for points that aren't as clearly divided. The argument that gene editing is unnatural is used as a negative, but the positive perspective points out that by saying something unnatural is bad, you imply that unnatural things are inherently bad. This would mean surely that medicine is bad, as it's man's invention. Conversely though, the argument that it is unnatural also poses the issue of playing God, which is a strong argument against for many people, possibly due to religious reasons. For example, the Roman Catholic Church believe that life begins at conception, and interfering with an embryo after this point is altering God's creation. Another argument in favour of editing is that germline editing is permanent, which means that if a genetic disease is cured in an individual, then the cure is passed on to subsequent generations, meaning that eventually genetic disease can be eradicated. The question is therefore raised that if you can make a disease-free human, why wouldn't you? However, as germline editing affects the genome permanently, an alteration to an individual's genome affects subsequent generations. Therefore, it could be argued that editing of the human genome is not a decision just to be made by the parents of the affected individual. This idea concerns many scientists, such as Francis Collins, the director of the National Institute of Health, who said, "Ethical issues are presented when the human genome is edited in such a way that affects subsequent generations." Furthermore, by saying that genetic disease needs curing, is admitting that there is something wrong with a person with a genetic disease, and they in some way need to be changed. Also, many are worried that even if the intentions behind genetically modified humans are good, it can still start us down a slippery slope. In 1999, Lee Silver, a biologist at Princeton University, wrote an article in Time magazine about the clinic of the future, in which designer babies could be created, where the parents could choose eye colour, etc. As said previously, CRISPR is currently labelled as the most promising area of gene editing in humans due to the simplicity and efficiency of design and potential for further research, not only at the germline level, but also at the local cellular level. In October 2016, at the University of West China, a group of oncologists used cells containing CRISPR to locally treat an adult patient with aggressive lung cancer. This means that any modification to the genome that are made will not be passed on to subsequent generations, and the adult is able to fully consent to the procedure. However, CRISPR is by no means perfect. One of the major criticisms of it are the off-target effects I mentioned earlier. Due to the guide sequences of CRISPR being short, there are often repeats of these sequences elsewhere other than the target regions of the genome. Therefore, CRISPR can have off-target effects by which non-target DNA is edited. However, there are algorithms such as eCRISP that are available so that off-target effects can be predicted. Furthermore, Dr. J. Keith Jung of the Massachusetts General Hospital says that off-target effects can be reduced to less than 1%, so it could be argued that the risk of is low enough to use CRISPR to eradicate a disease that is known to be very harmful. However, Jung also says that prediction algorithms are not as accurate as they could be, and for every 12 off-target effects that can be predicted, there can be up to 150 unknown effects. What if one of these off-target effects can disrupt a cancer suppressor gene? An example of this is the VEGF-A gene, which plays a large role in the growth of blood vessels, but overexpression of this gene is linked with many forms of cancer. If CRISPR is used to target the faulty form of this gene which resides on chromosome 6, the guide sequence used will also target regions on 22 other chromosomes. Is this worth the risk? Another question posed by germline editing of human embryos is that of the identity of the person once they are born. Would the person feel less close to their parent knowing that they do not share a gene and possibly a disease that the parent has? Or should the person even have been told? Would the person feel in some way different as a result of being genetically modified? Also, if all genetic disease can be eradicated, the population will become very homogeneous. Would this be a bad thing as there would be no genetic disease, or would it be too controlling of our own fate? These have been some of the big ethical issues surrounding gene editing in humans. I will now leave you to form your own opinions about how far science should progress with this, if at all. Thanks for watching.